Now let's take a look at another parameter that can tell us more about cell structure and function: fluorescence. As a brief review, fluorescence is a term used to describe the excitation of a fluorophore to a higher energy level, followed by the return of that fluorophore to its ground state with the emission of light. The energy in the emitted light is dependent on the energy level to which the fluorophore is excited, and that light has a specific wavelength and consequently a specific color. One of the most common ways to study cellular characteristics using flow cytometry involves the use of fluorescent molecules, such as fluorophore-labeled antibodies. In these experiments, the labeled antibody is added to the cell sample. The antibody then binds to a specific molecule on the cell surface or inside the cell. Finally, when laser light of the right wavelength strikes the fluorophore, a fluorescent signal is emitted and detected by the flow cytometer. How is this fluorescence information collected? The fluorescent light, coming from labeled cells as they pass through the laser, travels along the same path as the side scatter signal. As the light travels along this path, it is directed through a series of filters and mirrors so that particular wavelength ranges are delivered to the appropriate detectors. Fluorescence data is collected in generally the same way as forward and side scatter data. In a population of labeled cells, some will be brighter than others. As each cell crosses the path of the laser, a fluorescence signal is generated. The fluorescent light is then directed to the appropriate detector, where it is translated into a voltage pulse, proportional to the amount of fluorescence emitted. All of the voltage pulses are recorded and can be presented graphically. What if we want to do a two-color experiment? We need to look at the spectra of the two fluorophores to see if they are compatible. Alexafluor 488 and phycoerythrin, or RPE, are commonly used together. For these two fluorophores, 488 nanometer light is an efficient excitation source. When excited with 488 nanometer light, you can see that the emission peaks for these two dyes are far enough apart so that discrete emission data can be collected. Compatible dyes, such as these, allow scientists to easily detect two colors from a single laser. If we analyze data from the two-color experiment using a scatter plot, four distinct populations emerge. Looking at the dot plot in terms of quadrants, cells with only bright orange fluorescence appear in the upper left quadrant. Cells with only green fluorescence appear in the lower right quadrant. Cells with both bright green and bright orange fluorescence appear in the upper right quadrant. And finally, cells with both low green and low orange fluorescence appear in the lower left quadrant. Multiple fluorescence parameters are necessary to dissect complex biological systems. How does the flow cytometer collect discrete fluorescence data for the Alexafluor 488 and RPE fluorophores? Filters are available that can capture the peak fluorescence from each of these molecules. A 530 nanometer bandpass filter will collect most of the Alexafluor 488 peak, and a 585 nanometer bandpass filter will collect the bulk of the RPE peak. Using these filters, the proportional amounts of Alexafluor 488 and RPE fluorescence can be recorded for each cell. You can see that portions of each emission peak overlap one another. This is called spectral overlap, and we will talk about how to deal with this in the next tutorial. Let's take a moment to talk about filter nomenclature. Filters are normally defined by one of two parameters, either the center point for a bandpass filter or the cutoff point for a long or short pass filter. This is a typical bandpass filter specific for RPE. It has a center point of 585 nanometers and a width of 42 nanometers. So this filter optimally passes light in the wavelength range of 564 to 606 nanometers, which corresponds to the emission peak of RPE. Other filters used to resolve this peak include a 550 nanometer long pass filter and a 610 nanometer short pass filter. One final important point regarding data collection is the use of a threshold. If every single particle passing through the laser caused the instrument to collect data, the data pool would be dominated by information coming from a very large number of minute particles, like platelets and debris. To prevent this, a threshold is set such that a certain forward scatter pulse size must be exceeded for the instrument to collect the data. On the histogram, the blank area represents the small cells and debris that are excluded from analysis by the threshold. This means that the majority of events that the cytometer collects are the cells of interest. It is important to realize that the small particles are still passing through the instrument. They are just being ignored.